Hi physiology students, I hope you're all doing well. Welcome to Lab 5, and this is on cutaneous receptors. Much of these lab procedures in this lab could potentially be done in your own home. I won't make you do these procedures at home, but some of them, as you read through the procedures, you could mimic in your own home if you're looking to get a little more into this lab. I also posted a lab video from YouTube outlining or giving an example of people doing these same procedures in a lab setting. So feel free to watch that YouTube video as well. This lab is all about cutaneous sensation and also taste. So this ties in nicely with chapter 10, which is one of the chapters in this module that you'll be studying. Cutaneous sensations have to do with receptors in the cutaneous membrane, which is your skin. So these are all pain, cold, and heat receptors. They are naked dendrites, we call them, of sensory neurons. These touch and pressure receptors have special structures around their dendrites. They will be Merkel disc, Meissner's corpuscles, Pacinian corpuscles, or Ruffini corpuscles. And this kind of tells you the receptor type, the structure of them, and what they're specifically sensing. And the location of all of these receptors will be in the skin somewhere. So free nerve endings are all around hair follicles. The Merkel's discs are at the base of the epidermis. The Ruffinian corpuscles are deep in the dermis and the hypodermis, which is the layer of the fat underneath the skin. The Meissner's corpuscles are in the upper dermis, the papillary layer, and the Pacinian corpuscles are deeper in the dermis. And these are the sensations that each of these different nerves will help you with. So free nerve endings help with light touch, hot, cold. Nociception is another word for pain. Merkel's discs are associated with detecting sustained touch and indented depth. Ruffini corpuscles help detect skin stretch. And Meissner's corpuscles help detect changes in texture, slow vibrations, and Pacinian corpuscles help detect deeper pressures and fast vibrations. And this picture just shows you where in the skin these different nerve endings or nerve receptors will be located. And you can see here what they look like. You might have studied these a little bit in anatomy for what they look like, but each of these different cutaneous sensory receptors will sense something different. Whether it's texture, touch, pain, hot, cold, skin stretching, vibrations, deeper pressure, light brushing. And a lot of the procedures, if we were in person, would take us through kind of detecting these different sensory receptors and doing different stimuli for them. Cold receptors, there are many more receptors that respond to cold than to hot, and these are located close to the epidermis. They are stimulated by cold and also inhibited by warm. Some cold receptors also respond to menthol, which is a type of alcohol. The temperature range of response is from 8 to 28 degrees Celsius, and they also serve as an ion channel for sodium and calcium. So those are cold receptors. So there's many more cold receptors that will respond to cold than to hot. Warm receptors are located deeper in the dermis. They're excited by warming and inhibited by cooling. And they are different from receptors that detect painful heat. So that's something to keep in mind, that receptors that detect just warmth are completely different from receptors that might detect a burn or something painful in heat. Then we get to hot receptors, which is pain, experienced by a hot stimulus will be sensed by a special nociceptor, which is a receptor that has to do with pain, called a capsaicin receptor. They'll also serve as an ion channel, and they'll be a receptor for the chemical found in chili peppers, which kind of helps to give them their name. They will be activated at 43 degrees Celsius or higher. So think of these hot receptors associated with pain. Then pain receptors, nociceptors, another word for them, can be myelinated or unmyelinated. A sudden sharp pain will be transmitted by the myelinated neurons. Remember, things always travel faster through myelinated neurons and slower through unmyelinated neurons. So dull, persistent pain will be transmitted by unmyelinated neurons. Nociceptors may be activated by chemicals released by damaged tissues like ATP or by any sort of pH change or any other mechanical stimuli. Then an itch sensation, I don't believe the lab really goes through itch sensations, but an itch is usually stimulated by histamine release from mast cells and basophils. So it's sort of your body's way of a response of something foreign that it detects. A chronic itch is stimulated by other chemicals and does not respond to antihistamines. Receptors will stimulate in unmyelinated sensory axons to the spinal cord. So that's an itch sensation. A little bit about kind of the nerve and the nerve pathways. Sorry about this. I'm trying to change my view of the screen. Here we go. I have something in my line of sight that I'm trying to get out of from Zoom, and it won't let me here. So here a look at some of the pathways from heat, cold, and pain receptors. They're first carried into your spinal cord by thin myelinated and unmyelinated neurons to the lamina of the dorsal gray horns in the spinal cord. They'll synapse there, they'll cross sides, they'll desiccate, and they'll ascend the lateral spinothalamic tract, and then they'll synapse in your thalamus and continue on to the brain in the postcentral gyrus. More about pain. Emotional response to pain occurs when information is sent from the thalamus to the gyrus, that's part of the limbic system. And sometimes somatic pain information, which is from your skeletal muscles, can synapse on the same interneuron as a neuron carrying visceral pain information. The brain may interpret the heart pain as arm pain or the gallbladder as back pain. And this is called referred pain. So this is really common. If someone walks into your ER as a nurse, you want to keep in mind referred pain. It's pain that could be originating from an organ in the body, but it's referred to another part. So the heart attack is the most common. Heart pain is often referred to the entire left arm pain. The receptive field is this area of the skin that when stimulated changes the firing rate of a neuron. And the size of the receptive field depends on the density of receptors in that region of skin. So in your back, your legs, your neck, there are fewer receptors. So the receptive fields are extremely large. There are many receptors in your fingertips, so they're very sensitive to touch, and the receptive fields there are very small. The more receptors there are, the smaller the field, like your fingers, and the larger, and that gives them a larger area on the somatosensory cortex. A smaller receptive field is equal to greater tactile acuity or sharpness of sensation, like in your fingertips. The 2-point touch threshold test, which the YouTube video talks about and your procedure goes through, measures these receptive fields by seeing at what distance a person can perceive 2 separate points of touch. It measures tactile acuity, and this is really important in spacing dots in the Braille symbols, which is used for the blind, to help the blind read. So what is the 2-point touch threshold test? And if we were in lab, we would do these. So what we do is we start by taking an instrument with 2 sharp points and putting the points extremely close together, so close that when they touch your skin, it would feel like 1 point is touching your skin. And then we move them up 2 different points. And that's the area where you're detecting, you're perceiving 2 points of touch from the sensory neurons. So this is really important, like we said, in creating the raised dots in Braille, but it's also just important to look at different parts of the body to see where this 2-point threshold is. And again, the 2-point threshold refers to how far apart 2 points need to be until you feel that there's 2 of them. When they're extremely close together, it'll feel like just 1 point, but as you slowly move the 2 points apart, you'll start to begin to feel 2 points, and that's called the 2-point threshold. So we'll start at the low end. Right away, your first finger, your thumb, your big toe, the palm of your hand, they have the lowest 2-point threshold. And you can do this in lab. We would use an instrument called a caliper, but you could even use like a kid's scissors to do this. Don't cut yourselves, but If you slowly move the scissor points apart, you would just measure the length at when you could feel two separate points. And you'll notice in your fingers, especially your thumb or your digits, it would be 2 to 3 millimeters only when you could start to feel two separate points from the tips of a scissors. It would be much larger in your calf, your thigh, your back, your abdomen, your upper arm, even your forehead or the back of your neck. What 48 millimeters means, this means it takes about 48 millimeters for you to feel that there are 2 points being touched onto your skin from one instrument. And this just goes to show that the receptive field is different in your calf versus your first finger. So that's an example of one of the lab procedures that we would do, and the YouTube video should show that pretty well too. Lateral inhibition, these are receptors where touch is the strongest, and they'll be stimulated more than areas where touch is lighter. The receptors that are most strongly stimulated will inhibit those around them, and this allows us to perceive well-defined sensations at a single location instead of what we call a fuzzy border. This process will occur in the central nervous system, and this just shows how a blunt object touches the skin, and the stimulation, this shows the skin locations being stimulated, but the lateral inhibition sharpens perception. So your skin localizations of perceived touch will be greatest in the center of where that blunt object is touching. Then there's a little bit about taste in the lab where you would put different tastes on your tongue to determine where on your tongue and what type of taste your tongue can perceive. So this is a fun one to do in lab, again, that we can't do obviously, and you probably can't really mimic this at home. You can try if you can find some of the different taste sensations. But taste has to do with chemoreceptors, and these are interoreceptors that detect chemical changes within the body. Exteroreceptors detect changes from outside the body, which include taste and smell. So we're going to be focusing on exteroreceptors with taste. Taste responds to chemicals dissolved in food. Smell will respond to chemical molecules from the air. Olfaction, smelling, will greatly influence gustation for how you taste it. And if any of you have been to a winery, they usually tell you to smell the wine before you drink it to taste it. And that's just because olfaction, the sense of smell, will greatly influence your sense of taste. So taste is also called gustation. The receptors are called taste buds, and they consist of 50 to 100 specialized epithelial cells with long microvilli or extensions that will extend through the pore in the taste bud to the environment of the mouth. And this is what a taste bud will look like. On the tongue surface, you might have a taste pore, but here's the taste bud. There will be sensory nerve fibers attached to it, which will detect that sense of taste that a specific taste bud takes in. Taste buds are located in bumps on the tongue called papillae, and there's different types of papillae depending on where they're located. Fungiform papillae are found on the anterior surface of the tongue. Circumvallate papillae are found on the posterior surface, and foliate papillae are found on the sides of the tongue. The taste pathways, so the taste sensations will go into your facial and glossopharyngeal nerves, which are two cranial nerves. They'll travel up to the medulla oblongata, into the thalamus, into the primary gustatory cortex of the insula, which is a lobe in the brain. The taste cells of taste buds, they're specialized epithelial cells. They'll behave like neurons, so they'll be able to depolarize, producing action potentials. Cells release neurotransmitters onto sensory neurons, and the microvilli are what come into contact with the chemicals themselves. And what's unique about taste is that each taste bud has taste cells that are sensitive to a category of tastes. So there will be parts of the tongue that only can taste certain taste sensations. There's 5 categories of taste: salty, sour, sweet. Umami is like a meaty taste, and bitter no longer have certain areas for certain tastes. All regions of the tongue have taste buds for all the categories. So in essence, I told you that there were spaces of the tongue that are only specific to certain tastes, but there are technically all regions of the tongues that can taste every taste, just one might be smaller or stronger than the other. Taste will also be influenced by the temperature and the texture of the substance. So this just shows you the 5 categories of tastes and showing kind of how the taste itself, so this is sodium, so it would be a salty taste, will kind of be absorbed by those microvilli. It'll cause a depolarization in the taste bud cell, allowing calcium to release the neurotransmitter, and that neurotransmitter will send on that taste sensation through the sensory nerve. And this goes through the sour, the sweet, and umami, and the bitter. You don't need to know the differences with how each taste is perceived, but just it's good to understand how exactly you do taste by the salty, sour, sweet, umami, and bitter. So how exactly taste works? Salty, sodium will enter the taste cell, depolarize it. Sour, hydrogen ions will enter the cell and depolarize it. With sweet and umami, sugar or glutamate will bind to a receptor and activate a G protein and use the second messenger system. Bitter, quinine will bind to the receptor and activate the G protein second messenger system. And that's the end of this PowerPoint. For this, you should read through the procedures well. If you want to try some of the procedures at home, you're welcome to, especially the ones with touch, maybe not with taste, but the ones with touch and hot and cold, or trying to do the 2-point threshold test. And then you should answer the lab report questions as best you can. You might have to use other external sources. Feel free to use Google because there might be other references online to help you with some of this. But good luck. Please let me know if you have any questions, and I'll try to check the lab module discussion posts throughout the week. Thanks for listening, guys.